Well, Chris, it's great to see you again here at ANZUP 22. Have you enjoyed the meeting so far?
It's been a ripper, Lisa.
Hasn't it?
I've really enjoyed it, yeah.
Yeah, especially being back in person again has been amazing.
There is a lot to be said for that.
Yes, but we have worked you pretty hard this year as a organising committee. You've given plenty of talks, but I have to say, as a basic scientist, my favourite one was your one today, where you talked about how the sausage is made in respect to the ANZUP and ANZURAD trials, and I guess the importance of ANZUP in that. Did you want to tell me a bit more?
Yeah, absolutely. So let's just start with the notion that there are multiple ways to make a sausage, but in this case, conduct a clinical trial. And the challenge is us as investigators who see and treat patients to align with commercial interests of the pharmaceutical companies that make the drugs and finding that sweet spot. So the companies will sometimes run their own trials, which are very expensive, and they have to put a big bet down on whether that trial will get them to market and get the return on investment that they need to sustain, to make more drugs. So there's a very important goal they have. And we as investigators sometimes have goals that don't align with what a market-based drug development approach would be. But sometimes we, both the academic side and the pharmaceutical side, have a sweet spot where the projects are perfectly aligned. Pharma may not have the resources and the personnel to run a whole trial, but the cooperative groups can do that.
Right.
And back in 2013, we realised the potential sweet spot with the drug enzalutamide in metastatic hormone-sensitive prostate cancer, and recognising this has been recorded in 2022, so that's 9 years ago.
Wow.
So these sausages take a long time to make, and then when they're made, they're cured for a long time as well.
Yes.
So the goal is to really work out what is the value proposition where the cooperative groups like ANZUP can run high-level, high-quality trials that can be reviewed by regulatory agencies, support the development of a drug, and get an indication that may or may not be in the line of sight of a pharmaceutical company.
And it seems that something you didn't talk about in your talk, but certainly I've noticed in terms of the translational studies that are now coming out of ANZUP and ANZURAD, is the importance of collecting those very precious biological samples throughout the whole process. I mean, that seems like that's something pretty unique.
It is, very much so. So when you set up a trial, you actually have to think about what is the primary endpoint and how are you going to meet that. And the goal, I think, is how to get as much data out of that main data set that you're generating. And you have to put your best foot down to work out what may be relevant. So off the back of the Chartered study, we designed this blood collection, the tumour collection. Technology has advanced so we can marry the biological specimen analyses from different studies between Chartered, Stampede, and now ANZUP, and do really rigorous cross-study biomarker to define which patients benefit from which type of therapy, be it chemotherapy versus hormones versus both. But you're absolutely right, because we had the foresight and the academic ambition to have a broader lens of what we can get out of the trial, we did collect specimens. We also collected quality of life data in a much more granular format, and our quality of life data application manuscript is clearly very granular and provides a lot of information, both short-term and long-term, and complements the pharmaceutical sponsored study quality of life data from the other studies with abiraterone, enzalutamide, apalutamide, and darolutamide. So it all looks at the same problem but with a different spin, and we as investigator-initiated have a slightly more granular look at the data, which has been very informative. The other aspect is that we can then start to have all of this data in one database. For example, Lisa, here's a crazy one. So we now have the germline DNA, the general patient's DNA, of these men who present with metastatic disease.
Right.
What is it about their background, their basic DNA makeup, that had them present as their first presentation with cancer in their bones as opposed to a small amount of cancer in their
In the
prostate.
right.
So we now have the biological data, specimens to actually start analysing questions like that. But working with my colleague Anas Hamid and the Stampede team, Gert Attard, we've formed a group. We call it the Orthogonals.
Okay.
It sounds like a 1950s band.
It does a bit.
All up for the nerdy jokes. So this team will be able to analyse the germline DNA and see if it's associated with quality of life deterioration, if they have side effects from the chemotherapy or the hormonal therapy. Does the one gene expression profile match to a certain germline feature? And what is if a patient has a BRCA2 gene alteration, what's going on at the RNA level? So we get all of this data into one registry, and then we can do these orthogonal analyses across the dimensions, the patient's general DNA, their quality of life, their tumour specimen. So you're absolutely right. The investigator-sponsored trials can really be the gift that keeps on giving.
Yes, it's almost like the gold from this trial is still yet to come, right? It's beyond the actual trial outcomes in terms of the clinical outcomes. It's the scientific discoveries that are going to come from it just as exciting.
Absolutely. So we've got the skeleton, as you say, from the clinical trial result, and now we can layer on all the meat.
Yeah, it's very exciting. So do you see ANZUP sort of, continuing to play these sort of leading roles in some of these investigator-driven trials into the future?
For sure. So we leveraged the experience with Enzamet and Enzarad being the adjuvant study, with the notion that, yes, we can develop drugs in late-stage disease, but we're probably not going to cure it with one drug, one patient at a time. But if we bring these therapies earlier into the relapse from localized disease or prevent or delay resistance to hormonal therapy, then we may actually have more men die of something other than prostate cancer and decrease the death rate from prostate cancer. So we've set up that as a strategic goal within the ANZUP group, and some other groups have aligned with that. So with that led to the DAZZLE HiCap, where we're looking at darolutamide in the adjuvant setting with primary radiation or after surgery. Again, a hormonal therapy to decrease the relapse rate after prostate cancer treatments. But then we know the hormones work, we know the chemotherapies work. We have now got amazing technologies where we can look at thousands of genes at any one time. We can now image the tumours more specifically with PSMA PET imaging, FDG PET imaging. And what we need to do now is develop a strategy where we can be more specific with the next generations of drugs, the next generation of biomarkers as to which treatment a patient should get.
Right.
Would you like to hear about the Bespoke CAP trial?
I would love to.
So the Bespoke CAP trial is a concept right now. It's more of a platform, and Bespoke stands for biology specific, patient-oriented knowledge evolution. And the goal there is to get patients accrued to a registry. The main inclusion criteria will be, do they have metastatic prostate cancer on a conventional scan?
Okay.
And they get standard therapy. And we then map which patients got which therapy and for what reason by the investigators.
Right.
We can see which patients could potentially enroll on an escalation phase where they get intensified therapy based on a biomarker that matches to a drug that is out there for their tumor. And then we could also, with the patient-oriented, de-escalate for those who are doing really well and may not need an intensification strategy. So it's a phase 1 where we get an amazing amount of data, imaging data, quality of life data, social determinants data, what are the cultural barriers to getting patients onto therapy or access to therapies or onto clinical trials. So if we can get thousands of patients into that registry with the incredible amount of gene expression profile and exome data into one registry, we can really start to get an understanding who benefits from what therapy, who can access a given therapy and what is the barriers to therapy, but also be specific on who needs a more intense therapy versus a less intense therapy.
Intense, right. And so, I mean, you're going to be generating a huge amount of data. How do you manage all of that?
Pull a team together. So I really honestly see myself as a general contractor at this stage. And so what we're trying to do is we'll pull together teams from around the world. I hope I don't forget anyone. So the Medical Research Council and the Cancer Trials Ireland in the UK and Ireland, EORTC, the Spanish group SOGUC, the French group UniCancer and Peace Consortium, the Prostate Cancer Clinical Trials Consortium in America, ANZUP, of course, Canadian Cancer Trials Group in Canada. Canada gives it away where that comes from.
Yeah.
So we get everyone together and we bring many teams and we'll create working groups.
Yep.
One of the key working groups that I'm going to be working on, hopefully we can build it out of SAGENSI, the New Cancer Research Institute that we're the inaugural members of. Plug to the team, is that we are now going to try and see if we can work with the bioinformaticians and the Australian Institute of Machine Learning of getting all of that thousands, millions of data points into one registry, one repository, clinical and biological, and then do the machine learning.
Yes.
So we're bringing in not just biologists into this consortium, not just clinicians, not just patients and their patient advocates, but also new disciplines, bioinformaticians, machine learning, and bringing in new disciplines into fighting cancer.
Yeah, well, look, I think that's very exciting, and that's the future. I think it's been exciting to see the evolution of ANZUP towards that direction. So thanks so much for having this chat with me today. As I said, I really enjoyed that talk, and I think for a lot of people, whether they're clinical, scientific, basic scientific trainees and the like, all really got a lot out of hearing what's behind those really big papers and the journey that you take to get there. So thank you, Chris.
Thank you, Lisa, for the opportunity to share the bespoke CAP trial design.